Okay, everybody, we have a presentation today by our beloved member, Scott Keister. Scott is a Houston area transplant, joined our chapter 2 years ago or so.
Yes.
He's a very active member. He's a speaker at UNT Emeritus College. And an ardent, avid Leela sponsor. And I'm going to read a little dialogue on his little bio. And I'm reading it, thank you very much, Wanda, from the newsletter on my phone. Imagine that. Scott has been a certified Texas Master Naturalist since 2003. For 11 years, he's been a volunteer instructor for TWD's Texas Amphibian Watch, as well as U.S. FWL North American Amphibian Monitoring Project. For several years, he's worked with the Houston Bat Project as bridge host at the Wall Street Bridge Bat Colony. He has worked on different conservation-related projects and with conservation groups throughout Texas. Currently, he's active on our Speakers Bureau, and we're very grateful for that. He leads a monthly bird walk at Clear Creek Natural Heritage Center. He coordinates a lot of great stuff at Leela with the Amphibian Watch. He is a retired geologist and is finally getting close to finishing his MS in environmental science. He's a husband and partner to the very patient, lovely Deborah.
Yes. Very patient.
38 years. Can you imagine? And grandpa to 4 kids, 4 little grandkids: Hayden, Harper, Lorelei, and Annalise. And sometime toward the end, we already have that on there. Sometime it happened.
It happened. She was born, actually she was born 2 weeks early, much to her mother's delight, as you can imagine. And she's a healthy little baby. And unlike her big sister Lorelei, who, if you know who the Lorelei are, they are the Rhine maidens. They used to, in German myth, they were the ones that sang the song and drove the boats into the rocks. That's my granddaughter to a T. So it's very good that her little sister is a calm, sweet little thing.
Has anybody seen a commercial on TV for the Toyota RAV4, and it shows the car in the driveway, and they're loading up for all the activities they're going to do on the weekend, and the last little kid gets out and goes, "Herping!" Yeah, it was inspired by this guy. Scott Keister.
Thank you, Jan. Okay. Yet. I didn't get started. Yes, it worked. Hooray. I'm going to go back and forth to my notes a lot. Do I have to keep this thing?
Yeah, put it on your shirt.
Okay. Good God. I'm going to clank while I walk. All right. If you read the newsletter, you know that there are 3 species of leopard frogs that live here in the beautiful Metroplex. The Southern Plains and Rio Grande leopard frog. And they're all very close cousins, along with the rest of their friends in the Rana species complex. There are 21 recognized species of leopard frog. They vary. They range from northern Canada all the way to Panama. Why they don't jump the canal, I don't know, but apparently they didn't. They are not quite a cryptic species. There are ways to tell one leopard frog from the other. There is a true cryptic species in California that's a bunch of skinks, and I've seen pictures. You can't tell one from the other, and they live all around the Central Valley, and they don't breed with each other. They don't interbreed with each other, and they each are a separate species. We're close. Can we get rid of the row of lights that shines down right on the screen? If we get rid of that one, we're in good shape. Hey, look at that. All right. A species complex, you have to read this, is a situation where gene flow has been almost stopped between independent populations. But not enough time has passed since gene flow almost quit for there to be enough adaptations and morphological changes that you can easily tell one species from another. And looking at those pictures, you can tell that they're a little different, but they're also very, very much the same. And just to point out how much this has changed over the years, a first scientific description of a leopard frog is in 1782 by Johann Christian Daniel von Schreiber. Now, Johann was a German biologist, and almost all of his work was done with mammals. So far as I can tell from researching him, he never came to the United States, which was brand new those days. He didn't do anything else with leopard. We didn't do anything else with amphibians. He was a mammal guy. But somehow he gets credit for being the first person to describe a leopard frog. There's a story there somewhere, but I haven't found it yet. All right, on this graph, the solid line are the species names, the species themselves, that are recognized right now today. The dashed line are the different species that were recognized at that time, but have since gone by the wayside. So in 1900 to 1910, there were 16 recognized species of leopard frogs. 6 of those species names have been abandoned. They're either what are called synonyms, where you'll see them in parentheses at the end of a species name, or they're just not even used at all anymore. And long about then, people started doing different kinds of studies and looking at the breeding characteristics of these frogs. And they said, well, wait, a lot of these guys can hybridize with each other. They must be one species. And instead of 16 by 1940, we're down to 6 recognized species. And there's at least one herpetologist at the time that wanted to say they were all one big happy group. And then along came this thing called chromosome analysis. And then after chromosome analysis came DNA analysis. And the number of leopard frogs got up to where it is today at 21. When this was done in 1988, there were 20. Since then, a new one's been described. So this is a great example of lumpers versus splitters. Ever since the beginning of human beings categorizing things, there have been lumpers and splitters. Splitters want each separate little group to be their own selves. Lumpers want to say, okay, we can gather all these together because they're pretty much the same and they only have a little bit of variation. Now, at the moment, the splitters are in charge with the leopard frogs. As you saw, we have 21 different species. The opposite is happening with red poles. Red poles are little Arctic finches. They're cute little guys. They live in the Arctic. They fly across the Pacific and spend the summers in South America. When they were originally described in 1861, there were 8 different species described based on their appearance. Since then, the American Ornithological Union, who are the gods where this is concerned, have narrowed that down to 2, the common red pole and the hoary red pole. Common red poles have lots of brown streaking. Hoary red poles have a bright white breast. Until last year, when somebody said, oh, let's do a little DNA analysis and see what we really think. Sure enough, hoary red poles and common red poles have virtually identical DNA. They are a single species, despite the fact that there's a little bit of variation in their appearance. So in the red pole world, the lumper's got a good grip. In the leopard frog world, the splitter's got a good grip, at least at the moment. All right. What's next? Okay. So where are we right now with the leopard frogs? There are 21 species, like I said. And if you got that god-awful teal highlight, you live north of the Rio Grande. These are our 3 leopard frogs right here. And there are 3 lineages, A, B, and C. Not a lot of imagination. But new species have been added to this in recent times. Subaqua vocalis is a very close cousin to the Chiricahua leopard frog. However, he has very strange habits. If you got a little Latin in your background, you know that that means underwater voice, because these the males of this species call from about a meter under the water in the bottom of the pond. And their calls can barely be heard outside of the pond. This is very strange behavior for any frog. That advertisement call that the males give that you hear all the time is roughly the equivalent of, "Come to me, my darling, let us make beautiful tadpoles together. It will be wonderful." So it's very strange that this frog wants to call from underneath the water where it can barely be heard. And I'm sure there are graduate students from Arizona State out there running around right now trying to figure that out. The newest member of the crowd is the Atlantic Coast Leopard Frog, Rana. This was first described and accepted as a new species in 2012. It only got named last year. The range is centered on New York's Yankee Stadium. It includes northern New Jersey, parts of southeastern New York, and Staten Island. I was very disappointed. I was holding out for Rana Steinbrenner eye. For those of you who are sports fans, you get the joke. And no, they named it after one of the scientists who first decided that it might be a separate species. And it was actually pushed by citizen scientists, somebody who noticed the difference in the call and said, that's not a northern leopard frog, it's not a leopard, not a southern leopard frog, something else is going on here. And he happened to have enough clout with some locals that they started to do a little work in DNA. 5 universities, the New Jersey Department of Natural Resources, and a whole bunch of volunteers from the North American Amphibian Monitoring Project were involved in this. And one of the interesting things that came out of identifying this guy is that there's likely more unidentified species up and down I-95 between Massachusetts and North Carolina. And again, there's a bunch of graduate students out there somewhere scurrying around in ponds grabbing frogs to find out about that. All right, so what is all this? How does something like this get started? Way back when, 21,000 years ago, give or take a few, when Chicago looked a lot like this, minus the penguins in the foreground, and Kansas and Oklahoma looked a lot like this, and not just in the winter, all year long. And DFW looked about like this most of the year. In fact, it had a habitats and plants and animals very similar to the modern Canadian boreal forest. If you look at the map over here in the corner, that's the Laurentide ice sheet. Where is Ford when I need him to back me up? Down here is montane glaciation. So that's all ice and snow, even though it's not part of the ice sheet. This is tundra, just like we have in the Arctic. And then there's this little stretch of boreal forest that covers the Metroplex. South of that are temperate deciduous forests and grasslands and lots and lots and lots of animals. Because everybody who lived up here, slowly over the course of time as the glaciers move south along with the climate. And they're all hanging out down in here, and even all the way down to Panama, where the climate is about like what it is in Houston today. And they all hang out in little blobs of habitat that fit what they need to survive. Whether it's a tree, or grass, or a frog, or a mouse, or whatever, lots and lots of critters that lived in North America have moved down into the funnel at the bottom of the continent. And they live in things called glacial refugia. And the singular of refugia is not refuge, it's refugium. And it's my hope, more laughs, I want laughs at this one. Thank you. Maybe this was a preferred glacial refugium for frogs, maybe not. But the question is, just how many species were there at the time that the glaciers were at their maximum? And the bottom line is nobody really knows. Some people say as few as 3 or 4. Other people say as many as 8 or 12. The only consensus is a whole lot less than 21. So in the ensuing 21,000 years, that small number of species has become a much larger number of species. And that's what we're going to spend about the next 45 minutes talking about. And this comes down to, what is a species anyway? And you would think that this would be settled science. I mean, after all, this is the basis of biology. How do you describe things? What is a species? Well, it's still called the species problem because there's no real universal answer. And currently, I think I read something that said there are about 24 different species definitions kicking around in different parts of science right now. So where does this all get started? It actually gets started back with the caveman Oog and his wife Ulla. And Oog is out and he brings home his his rabbit and sets it down. And in the meantime, Alla has gone out and she's sorted out all the plants that she gathers. And she says, come here, Demi, I'm going to teach you what you shouldn't eat when you're out in the countryside. And she says, these are good to eat, these are not good to eat. And Og looks down, and he picks one out of the not good to eat pile, and he says, but honey, this one tastes good, and if I only eat a little bit of it, I don't get sick. I found that out. So I want to make another category. And Alla, being a lovely, good wife, she picks it up, puts it back in her category, smacks him, and says, listen, I'm trying to teach you something. That was the beginning of species. All right. So the modern species begins with that fellow there in the powdered wig. His buddies called him Carl, the taxman called him Mr. Linné, and the rest of us call him Carolus Linnaeus. He is the one who came up with the binomial system for naming organisms that we still use today, genus and species. It's his fault that we all had to remember, what is it, divine King Philip called out for great Scott. Anyway, when Carl was a botanist, and he went around and described plants by their sexual organs, their flowers. How many pistils, how many stamens, how many anthers, and so on. Is the ovary superior or inferior to the flower, and all that kind of stuff. And other folks went out and started using the same system for all kinds of things, even rocks. So this was a big deal in the early 1700s that Carl came up with this, and everyone adopted it, and we still use it today. But the species he talked about were morphological or typological species, how something looks and just how it looks. Maybe you take the skin off and look at the muscles and bones, but it's still based on morphology. So those are morphological or typological species. And this works pretty well. However, sometimes you have a flower that has the same number of pistils and stamens and the ovary in the same place that's pink, and sometimes you have one that's yellow. Are these two different species, or are they subspecies of the same organism? And along comes the concept of the polytypic species, to where you can have a certain amount of variation within a species, but it's still based on morphology. This is how things sit until the beginning of the 20th century. Lots and lots of other stuff is going on, but no one's ever come up with anything that really synthesizes more information than old Carl's original system. Now, in the beginning of the 20th century, people started talking about, well, what about their paleontology? What about their ancestry? What about their ecology? What about their cytology? What about their cells? All of these things have to be taken into account when you look at an organism and ask yourself, what is it? And Ernst Mayr, who is a German biologist in the early 40s, wrote a very influential book where he introduced the biological species concept. And the biological species concept revolves around four things. A species is a group of organisms that are morphologically similar, that apparently share the same ancestry, that produce viable offspring, and most importantly, are reproductively isolated from other groups. This was the thing that Ernst Mayr really introduced to the system, was reproductive isolation. Along with Ernst, there were a bunch of other folks, and one of the other giants of 20th century biology is Theodosius Dobzhansky, and he was a Ukrainian biologist who worked in the United States. And between them, either working in parallel or working together, Mayr and Dobzhansky are given credit for the modern biological synthesis, which is where you take not just morphology, but all these other things into account when you try and describe an individual species. So you look at all kinds of things, not just how the critter is built, to determine what its relationships are to other groups, and is it a separate group, or is it part of is it one member of a larger group? And even with all of that, it's still not where it's perfect. And this quote from Dobzhansky is one of my favorites. Of late, the futility of attempts to find a universally valid criterion for distinguishing species has come to be fairly generally, if reluctantly, recognized. This man literally was a giant of science. And after all the work has been done, he says, we still don't have it quite right, and we still don't know quite what we need to do to make it right. So, a species complex comes out of this concept. These are closely related species that nonetheless are separate. Even though they're separate, they still haven't been separate long enough to develop a whole lot of variation. Another species complex that just kind of tickles me a little bit, this is a sidebar, is the genetic species. This has nothing to do with macro critters. This is bacteria. Bacteria, single-cell little beasties. They have no nucleus. Their DNA is wrapped around their cell walls. They are the most primitive form of life. Every once in a while, bacteria A and bacteria B, they get together and they kind of bump into each other. And for various physical and... Stop yawning, Jan. You're not giving me any help here. I know. For various physical and biochemical reasons, their cell walls kind of soften up and become porous. And a little bit of DNA from A goes to B, and a little bit of DNA goes from B goes to A. And then they jostle apart, and their cell walls harden back up. What do you have? Do you have a subspecies of A with a little bit of B? Do you have a subspecies of B with a little bit of A? Or do you have two whole new species? Horizontal gene transfer is what that's called. And it drives people nuts when they're trying to categorize things, particularly microscopic things. And I think that that is fascinating, and I kind of just watch it as a sideline. Okay. All right, everybody, jump back in the DeLorean with Doc and Marty. A quick trip down memory lane to high school biology class. As you will recall, when you make a gamete, an egg or a sperm cell, you do this thing called meiosis, where the chromosomes duplicate themselves, and then they split apart, but before they split apart, they kind of share little bits with each other. So some of mom's chromosome goes to dad, some of dad's chromosome goes to mom, and each generation of us has a unique genetic character because of that inborn variation in the system. That's recombination. When you hear about recombinant DNA, that's what they're talking about, splicing genes back and forth, and independent assortment, where you can end up with some of mom's chromosomes and some of dad's chromosomes in each different gamete. Along with that, while these guys are duplicating, all the G's, T's, C's, and A's in the DNA don't always go back together exactly the same way. There are little variations in there. Those are called mutations. And all of this is genetic variation. That is the beginning of the formation of new species. It is also responsible for what is called genotypic plasticity. There's a lot of variation in the genes of any critter. You don't believe me? Stand Larry or RJ next to Dave Ford. There's variation there. You know, within the genome of human beings, there's lots of variation. So this genetic variation is one half of what you need to create new species. The other half is selective pressure. Now, selective pressure decides which of those mutations is adaptive and which is maladaptive. Almost all, are completely neutral. They make no difference to how well an organism lives their lives, how many kids they have, or anything else. Every once in a while, one of them comes along that's adaptive based on the environment in which that critter lives. That's selective pressure. From the world of frogs, let's go back to our frogs, and we're hopping north, doing our thing. Got to find a new pond, less competition, more room to make tads, hop. And they get to the southern tier of the United States, which is now all nice and deciduous forest and full of rain and ponds and lots of great places for frogs to live. Some of those frogs follow Horace Greeley's example, and they go west, young frog, hop, off to find new ponds. This is all well and good right now, and this is several thousand years ago, because there's lots of rain and lots of ponds and everything's cool. So the frogs that hop into those ponds, they have a little bit of variation in how long their tads stay tads. Some of them are 60-day tads, some of them are 120-day tads, and a few in between. The 60-day tads, they get out of the pond sooner. but they're small. They don't compete as well with the other frogs around because they're not as well developed. They're not as big. And they're also not as good as getting out of the way of Great Uncle Henry, who would like to have them for breakfast. So they're okay, they're surviving, but the 120-day tads, they're bigger, they're stronger. When they metamorphose, they stand a better chance of surviving and passing their genes on to the next generation. So while this is going on, the 120-day tads tend to dominate in the population. They have more babies, their babies have more babies, and eventually the 60-day tads are this sort of little lineage off on the side, and the 120-day tads are the majority of the population. That's called, when that happens, that gene is called fixed in the population, when almost everybody has it. This works real well as long as everything is damp and moist and there's lots of rain. Now, as things get warmer in the western United States, they also get drier. So a lot of those nice permanent ponds flat disappear. A lot of them become seasonal. Many of them become ephemeral. They only have water in them for a little while each season. Now, all of a sudden, those 60-day tads, they got the upper hand because they get out of the pond quicker. They get out, they hop off into the underbrush and they hide on the shoreline. The 120-day tads are still swimming around in a pond that's getting smaller and smaller and smaller. And every heron and egret for miles around is going, manna from heaven, come let's eat. And the 120-day tads, they don't pass on their genes so much anymore. So the selective pressure has changed to favor the 60-day tads. That's an example of genetic variation and selective pressure. That's the mechanism of speciation, always. Because each different critter has to adapt to the environment in which they live. And with enough adaptation, you become different from other species, other members that were in your original group. So there are 4 pathways to speciation: Allopatric, Peripatric, Parapatric, and Sympatric. These are not 4-lane divided limited access highways. They're ruts in a dirt road. And you can sort of go back and forth in between them, and they share a lot of characteristics. Essentially, you start out with a single population. Something happens that population becomes separated into 2 groups, or more than 2 groups. Over time, genetics does its thing, selective pressure is out there, and the groups become reproductively isolated. At that point, they are separate species. Okay, allopatric speciation is my personal favorite because it's brute force and it requires geology. Yay! And the poster child for allopatric species are the ratites, the big flightless birds. About 152 million years ago, South America, Africa, India, Australia, and Antarctica were all jammed together into a giant continent called Gondwana. Great name. I don't know where it came from. And roaming around on Gondwana is the ancestor of the ratites. You may call him the proto-ratite, him or her the proto-ratite if you wish. But apparently it lived all over the place. And over time, as you know, these guys all separated by rather large geographic barriers known as oceans. And they took their proto-ratite ancestor with them. And over time, those proto-ratite ancestors responded with genetic variation and selective pressure and became separate species. However, these guys are all still very closely related. They are called ratites because they do not have the keel bone that other birds have. Even if there was a genetic mutation and one of them grew big wings, they couldn't fly because they have nothing to attach flight muscles to. Now, in Africa, there is one ratite, the ostrich. In northern Australia and New Guinea, there are 6 species of cassowary. In New Zealand, there were moas, there aren't anymore, and there are still 6 species of kiwi. In the rest of Australia, there are 2 species of emus. In South America, there are 2 ratites and 21 tintamus. This is what happens when you start out with an ancestor and you break the area apart. You don't have to have something as dramatic as an ocean forming in between. You could have a change in climate that forms a desert. You could have a mountain range come up in the middle of a population. The important thing here is that you had a large population and it was broken into several pieces by geologic processes. That's allopatric speciation. Second cousin to allopatric speciation is parapatric speciation. And the poster children here are the American brown bear, otherwise known as the grizzly, and the polar bear. This is almost like allopatric speciation, except there's no definite break. There's no mountain range, there's no ocean, there's no giant river, no new desert, anything. It's just a matter of a small population moves away from the main population to a new area, and they begin to adapt to their new environment. So over about 4 or 5 million years, every once in a while, a few brown bears would wander down to the coast. And they would join the other brown bears that had wandered down to the coast. And over time, they adapted to the Arctic sea lifestyle. And now we have polar bears and grizzly bears. And they still can crossbreed on those situations where they come across each other. You may have read about ghost bears. Ghost bears are hybrid grizzly bears and polar bears. So you start out with a single population. The initial step is somebody moves out to a new niche looking for more food or more room. Not everybody goes, but a few go, and they continue to go over time. But the main population stays back in the original territory. Genetics and selective pressure does its thing, and eventually you end up with two separate species that have their own ranges. If you want to think about this in terms of the frogs, here we have our main body of frogs, hopping north, heading out of Mexico into the United States. And as they're hopping up, a mountain valley opens up to one side of the main migration route. A few of them hop up into that mountain valley. And they find some ponds, and there's not many frogs there for competition, so it's a good place. They stay, and they survive. And they pass on their genes from one generation to the next. As you hop up into a mountain valley, things are cooler. Within the frog's genome is the ability to, some of them can stand cooler weather. They're more active even when it's cold. Those are the ones that survive and pass on their genes to their offspring, who pass it on to their offspring. Given enough time and enough generations, you end up with mountain valley frogs and main population frogs. Now you have two separate species, and even if you take a mountain valley frog down and stick it with a main population, it's probably not going to survive, and vice versa. That would be a way to look at it in terms of our frog friends. The next one is Peripatric speciation. We have a big difference here. There is no longer a geographic barrier between the groups. They actually bump up against each other and overlap. And the poster children here are the Larus gulls, which are actually what are called ring species. But basically, the European blackback gull lives in here. It can crossbreed with the Siberian blackback gull, which is its nearest neighbor in going around the ring. But it can't necessarily crossbreed with Huglin's gull, which is over here in number 3, although the Siberian can. Part of that is geographic separation. Even though the ranges of each species overlap with the neighbors, it's too far for these guys to fly to mate with these guys. So they have become definite separate species with some geographic isolation. And you work your way around the ring from the, Huglin's gull, to Berula's gull, to Vega's gull, to the American herring gull, to the European herring gull. The European herring gull, even though it looks almost exactly like the European blackback gull, they don't even talk to each other, let alone crossbreed. And it's because the species, the actual ring started somewhere in here. Some went this way, some went that way. But there was a long enough separation right here that these two do not crossbreed at all. But in between, you've got your niches still in touch with each other, separated enough that you get reproductive isolation, but not completely separated. That's Parapatric speciation. The last pathway is Sympatric speciation. This one is very controversial. There are a lot of biologists who say Sympatric speciation is just a single subset of Parapatric speciation and shouldn't even exist. The poster children here are the cichlid fish of Lake Victoria in Africa. If you've ever wandered through PetSmart and gone to the tropical fish part, there are always bunches of cichlids there with different colors and patterns and all of that. There are 800 different species of cichlid fish in Lake Victoria, and they all have a single common ancestor. Now, Lake Victoria, right down in here, is part of the East African Rift. Sometimes it has water and sometimes it dries up. The last time it dried up was 17,300 years ago, give or take. So in the ensuing 17,000 years, one fish became 800 different fish. Now, how this happens is a lot based on behavior and habitat choice. Some cichlids live close to the shore in the rocks. Some live close to the shore where it's sandy. Some live close to the shore where there's lots of vegetation. Some live further down in the water column, but they still stay close to the bottom. Some live all the way at the bottom of the lake. Others live in the middle of the water column and float around in the center. Others live at the top of the column and float around in the center. It is their behavior and their habitat choice that has kept them reproductively isolated. And that's why this is controversial, because this is just one big fish bowl. Why don't they interbreed with each other? They certainly can, but they don't because of their habitat and behavioral differences. So that's what Sympatric speciation revolves around, is the concept of habitat choice and the concept of behavioral differences molding that habitat choice. Okay, those are the four different pathways to speciation. How do they stay separate and how do they get separate? There are several mechanisms that help species diverge and stay separated. Prezygotic mechanisms may be thought of those as before making whoopee. Postzygotic mechanisms are the sins of the parents visited on the offspring. Prezygotic mechanisms, a very important one with our frog friends, is temporal isolation, breeding at different times of the year. You don't crossbreed if you do all your breeding or most of it in the spring and another species that you share territory with does it in the fall. Temporal isolation of breeding. Behavioral isolation, different courting rituals. All these frogs have different, slightly different calls. And studies have shown very, very clearly that females are drawn only to the mating calls of their conspecific males, so the males in their species. So behavioral isolation is also important with the frogs. Mechanical isolation, differing habitats. There's a leopard frog that likes to live in streams and leopard frogs that like to live in ponds. Different habitats, isolation. Isolation leads to eventual speciation and change. And then there's gametic isolation, which has nothing to do with frogs, but it's kind of interesting anyway. Just about everybody has seen Sir David Attenborough or someone from the National Geographic Society talk about how on some magic night every spring when the moon's just right, every reef-living coral in the tropics tosses their sperm and eggs to the sea with grand abandon. This would not work except for the fact that the ova from coral A and the sperm from coral B cannot fuse to make a zygote. They aren't the right shape. So they bump into each other and they wander off and do their thing. And along comes sperm from A and ova from A. They fuse, make a zygote. The zygote floats around for a while and becomes a free-swimming larvae. And almost all of them are eaten by zooplankton. And the ones that aren't eaten, a very small number eventually land on just the right substrate and they grow into new corals. But that's why they do that. They throw out lots and lots and lots of gametes because they're a favorite food of lots of critters. Postzygotic mechanisms come in three categories. Zygote mortality. You're so sickly that you don't even get born. So you never get a chance to pass along your genes to anybody else. Non-viable hybrids. You're so sickly that you don't make adulthood. Or my personal favorite, you're strong enough and healthy enough to make it to adulthood, but you're not like dad's side of the family and you're not like mom's side of the family and no one wants to mate with you. They all look at you and go, "You ugly, and your mama dresses you funny, and I am not going to mate with you." That is a non-viable hybrid, and that happens a lot with frogs. Lots of breeding studies have been done, and they show that hybrids tend to be less able to breed than those that stay pure to their species. And lastly is hybrid sterility. There's also some hybrid sterility amongst frogs. All of you who are horse folks, you know what happens when you breed your quarter horse to the neighbor's burro. You get a mule, the absolute definition of a sterile hybrid. Grows up, good, pulls the plow, whatever else, can't have kids, screwed up genes. Hybrid sterility. So those are the isolation mechanisms that help species diverge and that keep them separate. And we'll talk about how some of those affect our local frogs. For those of you who have been waiting patiently for how to tell the local frogs apart, we're there, folks. Hallelujah, everybody says. Okay, we have 3 species of leopard frogs. The southern leopard frog, Rana sphenocephala, the plains leopard frog, Rana blairi, and the Rio Grande leopard frog, Rana berlandieri. If you look down here at their range maps, you see that we're right on the edge of the southern leopard frog range. We're sort of down towards the bottom of the plains leopard frog range, and we're right in this odd little peak here in the Rio Grande leopard frogs range. If you look at it on a map, you can see the different colors and where they overlap. In 1981, when the original studies were done, there were 4 counties in which all 3 species were found. Basque or Bosque? Somebody who's a native Texan. Bosque. Johnson, McLennan, and Somerville. And that was in 1981 when this original map was made and the studies were done. In 1998, the Parks and Wildlife Department went out and did some censusing, and they discovered that the Rio Grande leopard frog had moved north. So they're expanding their territory towards the Red River. And 4 more counties had been added to the overlap of all 3 species. Tarrant, Dallas, Collin, and most important, Denton. Yay for us. So what we're looking at when we look at these 3 species is we're looking at the expansion of the range of the Rio Grande leopard frog. The plains leopard frog and southern leopard frog have probably overlapped for a long time. But the plains leopard frog is the new kid on the block. How to tell them apart? Every leopard frog, in fact every true frog, has what is called a dorsolateral or epithelial fold. This is a ridge of skin that runs from behind their eye down to their hip bone. It is important in field ID. Southern leopard frog's epithelial fold is entire with no break and no offset. Plains leopard frogs and Rio Grande leopard frogs are broken and there is an offset. That is how you start to tell them apart. There are a couple of other things you look for, and I will talk about those in a little more detail. I need my notes now so I don't forget anything. So bear with me for a sec while I get to page 23. All right. The southern leopard frog. This, by the way, is probably the most populous leopard frog in the area. Their dorsolateral fold is continuous, narrow, conspicuously raised, and very light colored. It's almost white. All true frogs have what is called a tympanium. It's a little round flap of skin that sits exactly over their eardrum. On the southern leopard frog, the tympanium has a brilliant white spot, 99-point-something% of the time. Occasionally one comes along where that little bit of phenotypic plasticity has left it without a good white spot on its tympanium, but most of the time it's there. They also have a very prominent supralabial stripe, is what that is called. It's another ridge of skin that sits right above their upper lip. On southern leopard frogs, it is very similar in character to the dorsolateral fold. It's narrow, raised, and very light colored. Southern leopard frogs have huge balloon-like vocal sacs, which are usually pigmented and possibly a little darker during the breeding season. You can see a picture of him over here. As far as their coloration is concerned, all leopard frogs are brown, varying to darker shades of green or combinations of the two. definite white lobe. 2 or 3 irregular rows of dark, round spots on the dorsum, that's the back, between light dorsal stripes and on the sides of the body. The spots are often outlined by a lighter color, which you can see on some of these. These guys live, as I said, pretty much where the western edge. They stretch all the way around the east coast and all the way back up to New York. So they are a very populous frog. Behavior. All leopard frogs do the same thing to escape predators. They often sit on the side of a pond rather than in the water during the day when they're hiding out from the sun. If you disturb them, they will go eep at the top of their lungs and jump in the water. There are several other frogs, in fact most other frogs will jump in the water when you walk up on them, but only leopard frogs go eep while they're doing it, and that is exactly what it sounds like. I don't need to record it, that's it. So if you walk up on a pond and a frog jumps in the water and it eeps at you, at least you know it's a leopard frog. You can start from there. If it's a little bitty frog and a whole bunch of them jump in all at once, it's a cricket frog. You'll never catch them. They're tiny little guys. All right, they have a major breeding period in late winter and early spring, but occasionally, rarely, pretty much all year long. The males call from about an hour after sunset until dawn. Oh, by the way, the scientific name, Rana sphenocephala, comes from 2 Greek words, sphenos meaning wedge and cephalos meaning head, because these guys have got kind of a pointed snout. So it looks to their morphology to define the name. They prefer permanent freshwater habitats with still, clear water, although they will, near the coast, breed in brackish water. Now we got to find out if I can play the call here. Hopefully this will happen. Yay! Hear the laugh? That's diagnostic, that laughing sound. Like 5 or 6 in a row. All right, that's the southern leopard frog. When you hear it and you listen for a while, it becomes very distinct. That laugh sound is what you're looking for, what you're listening for. The old-timey name for these guys was the laughing frog, because they have that great repertoire of calls, and it's punctuated by that which a lot of people equaled to laughter. Okay, our other friend is the Rio Grande leopard frog, the new kid on the block. These are becoming more common. I'm pretty sure we heard one at Lila. I am absolutely sure I have heard them at the Fort Worth Nature Center. Positively. The Rio Grande leopard frog, Rana berlandieri, is named in honor of Jean-Louis Berlandieri, a French naturalist who worked for the Mexican government surveying wildlife in Texas in the late 1820s. And he was the first person to describe the species. All leopard frogs are about the same size. Adults run 3 to 3.5, 4 inches long. And that's from nose to vent, not counting legs. So they're all about the same size. This is another smooth skin, angular head. The dorsolateral fold is broken and displaced medially, as they say, which means it's moved in towards the back. So that's one thing to look for. The second thing to look for is the tympanium spot is very dull and oftentimes not there at all. Whoop. Yeah. What'd they do? Jonathan! Ah. Okay. Sorry, folks.
Can I have the microphone real quick?
Sure.
Just talk loud for a few minutes.
Talk loud. That's never a problem. Okay, back to the rear grand leopard frog. The dorsolateral fold not only is broken, it is also less well defined. You can see this is much less of that tight, high ridge than on the southern leopard frog. The supralabial stripe likewise is not as well defined. It's more of a ridge than a bump. It's more of a little bump than an actual ridge. But the important thing is to look for that break in the fold. Now the vocal sac is smaller, less conspicuous, and it's always dark. On the real grand, on the southern leopard frog, if you remember the picture, he looked white on his underside. They darken up during the breeding season. These guys, the underside of their jaw, which I couldn't get a picture of, is always a little darker. It's always kind of gray rather than white. And general distribution, these guys are found in Texas and northeastern Mexico. Behavior, Rana berlandii is nocturnal. All of them are nocturnal pretty much. But it burrows into the dirt during the day to hide from the heat and the sun. This is probably in response to the fact that they live mostly in South Texas and northeastern Mexico, where you got to get out of the sun and underneath something or you're going to dry out. When you're a frog, your life revolves around two things: getting enough food to eat and staying damp. And you have to run a balance in between those two. And their response to this is to dig themselves into the dirt during the day when it's hot, which neither of the other two species do. So if you are out there and wandering along and you find one that lives, and you find one, and he digs himself out as he's trying to get away, you know you have a little hint that it might be a real grand leopard frog. The other hint is that these guys live and breed in moving water. They're not pond frogs, they're creek frogs. And they like it that way. So they tend to stay in places where there are moving water. I'm pretty sure, like I said, we heard one once in the Elm Fork on a night out at Leela. Okay, and let's, all right.
It should be good.
Okay.
Hopefully.
Hopefully. Hopefully it's good. Mm-hmm.
We're still new to this system, so bear with us for a few technical difficulties from time to time.
Okay. All right. Am I mic'd? Is there anything else I need to do with the Rio Grande leopard frog? Yes, I need to play their call. Now, the call of the Rio Grande leopard frog, I need more. Can we do volume? Do we know how to do volume? Awesome. Thank you, Jonathan. In the Rio Grande leopard frog, it's not the laugh that's prominent, it's the growl. The laugh is kind of a lower pitched background noise, and it doesn't have that ah. But the growl is what you want to hear. If you listen to a leopard frog, and that's me messing with the microphone. I thought I'd cut that out of there. Okay, anyway. So the call here, which is what you want to think about, is more of a growl than a laugh. And that's the way you tell them when you're sitting out at Bittern Marsh listening at night. That's how you tell that that's a Rio Grande leopard frog, because they growl. All right. Now these are tadpoles of Rio Grande leopard frogs over here on your left, and southern leopard frogs on the right. You notice the head is larger, the body's larger, and there's much more muscular development in the tail. Adaptations to living in a running water environment. You got to be able to fight the current every once in a while when you're a Rio Grande leopard frog tadpole. So they have adapted to their environment. Okay, last member of the group, the plains leopard frog, Rana blairi. W. Frank Blair was a UT herpetologist who pioneered the study of systematics and evolutionary biology of frogs. So they named this frog after him. Again, we have the very definite break in the dorsolateral fold. However, Like the southern leopard frog, it's a very nice, distinct, raised ridge that's easy to see, and it's also bright whitely colored. Same thing for the supralabial line. It looks very much like a southern leopard frog, and it even has the white compendium spot of the southern leopard frog. The difference is the break in the dorsolateral fold. There's also a difference in habitat. These guys prefer to breed and live in still water, but they're perfectly happy in grassland environments. Southern leopard frogs kind of need a swamp. Plains leopard frogs, as benefits their name, are perfectly happy in a big mud puddle out in the grass because they have a very short larval period for their tadpoles. So there's some habitat separation there. It's not that plains leopard frogs won't breed in the swamp where they have lots of water. It's just that they're also comfortable and maybe more comfortable breeding off in the grasslands in a good-sized puddle. See what else? Reproduction peaks in early spring in April, but occasionally occurs year-round with warm rains. These guys are found in kind of a big arc from western Illinois on down into North Texas. As you see, we're sort of the southern edge of their range when you look at the range map. Let's see, anything else about plains leopard frogs? Nope. Okay, so those are our 3 leopard frogs. We'll come back. There'll be a quiz about the calls, so I have to play the call. Have to remember to do that. All right, and this one's a little different still. That was interesting. When you hear the plains leopard frog, it sounds like that. But it's more static than the laugh of the southern leopard frog. It's da-da-da, 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 groups of 3. And they don't growl very much, despite what you were hearing there. They don't make a whole lot of growl noise. It's da-da-da, da-da-da, da-da-da. And it's, after you listen for a while, all 3 will become distinctive. But it takes a little bit of sitting there and going, okay, was it 6 or was it 3? Did it growl? Did it laugh? Did it just chuckle? What? To kind of figure these things out. Now, in the areas where these 3 overlap, how do they stay separate? And that's what we're going to finish up with, is the isolation of these different species. In areas where there are separate populations, this is Rio Grande leopard frog down in Llano and Gillespie counties where there's only Rio Grande leopard frogs. They have a big breeding peak in the spring and a second big breeding peak in the fall. The plains leopard frog in central Oklahoma, where there are only plains leopard frogs, have a single breeding peak in April and May. And the southern leopard frog in the piney woods has a big peak in the spring and a little one in the fall. Now, in the 8 counties where the 3 of them live together, they have isolated their breeding periods. And there's a lot of discussion about how this happens. Probably this happens because of competition for resources amongst young adult frogs, because they're already sort of separated by habitat. So the tads don't really overlap with one another very much. Some are in the rivers, some are in the little ponds, some are in the big ponds. So it has to be something to do with competition among adults that, back. There we go. Okay, it has to be something to do with competition for resources that they have sorted themselves out into these 3 very separate breeding periods. And there's people working on that, but nobody's come up and said, this is why this happens. But it does happen. It's very well documented. And most researchers think that this is the biggest reason that they maintain separate species identity without a lot of hybridizing in areas where they're sympatric, where they share the same range. The other is habitat isolation. Rio Grande leopard frogs prefer to live and breed in running water. Southern leopard frogs and plains leopard frogs like standing water. Southern leopard frogs, being southern gentlemen and ladies, they want a big old pond. Plains leopard frogs, being pioneer species, they're happy with a pond out in the grass. So there is some habitat isolation as well as temporal isolation. And lastly, there is behavioral isolation. That, by the way, is amplexus. She's down here, he's up there. As I said earlier, female frogs are very, very picky about what call they will respond to come breeding time. They really will only respond to the call of their own species. They don't, and this is also a very large part of reproductive isolation, is that fidelity to the mating call of the females. So a female plains leopard frog will wait until she hears a male plains leopard frog before she goes and engages in amplexus and lays a bunch of eggs in the wood, in the pond. Oh, by the way, all leopard frog eggs are laid in masses, a big gob, as opposed to toads who tend to lay them in long, sticky strings that clog up the filter in your pond in the backyard. And they generally lay them around a piece of vegetation so that it kind of stays in place. Oh, I forgot one other thing. Behavioral isolation, still there, good. Rio Grande leopard frogs tend to call from the shoreline. Even Rio Grande leopard frogs that decide they're going to leave the creek and move to the pond, they still call from the shoreline. So not only do they have a unique call, they have a unique location. Southern leopard frogs tend to call from floating out in the middle of the pond, but they call from almost completely submerged. All you see when you see a male southern leopard frog calling out in a pond, you see his head pop up, the vocal sacs come out, the head pops up, the vocal sacs collapse, and he falls back down, and he slides back down in the water. A plains leopard frog tends to call from the same area, but he's going to hang on to a floating piece of vegetation, so he can keep his head up above water the whole time. Another behavioral difference that allows them to stay isolated. So, that's the show, folks. For further reading, if you want to learn more about how North America was repopulated after the most recent glaciation, and it was the most recent, there have been 7 in about the last million years or so. There have been 7 different glacial episodes, and this has happened each time. This is a neat book. It talks not just about frogs, but about all kinds of mammals and grasses and plants and trees and birds and everybody else. And it shows you how the different species repopulated the continent. If you just can't get enough about frogs, if you've got to know everything there is to know about the frogs, at least the ones that live north of the Rio Grande, Frogs of the United States and Canada by Ken Dodd, is a wonderful reference. Go to the library. It's $120 a piece for each volume. So that is not for the faint of heart. That was my Christmas present. $240 worth of books on frogs. I got new binoculars for my birthday. I'm not complaining. If you just want a really good field guide to the amphibians of Texas, this is it. Texas Amphibians. It's part of the Texas Natural History Guide series put out by the University of Texas at Austin. And it encapsulates a great deal of this information down to bullet points and short sentences so that you get a lot of it without having to worry about how many times different hybrids have been reported in this place or that place. So I recommend that as a field guide. It's got great pictures. It covers not just frogs, but toads and salamanders, of which there are plenty in the state of Texas. Credit should be given where credit is due. This is Dave Hillis. He is, as you see, the Alfred W. Rourke Centennial Professor of the Natural Sciences down there with the Orange Bloods. When I put together the bones of this talk last fall to be a part of a Texas Amphibian Watch training at the Fort Worth Nature Center, I started researching articles, and Dave's name comes up a lot whenever you talk about frogs, particularly leopard frogs. I tracked him down, I sent him an email, explained what I was doing, and politely asked if he could suggest 1 or 2 papers that I could get off of Google Scholar to sort of, flesh out my information. He sent me 11 papers and said, here, everything you need to know is in here. Have fun. Good luck. And most of the science you heard this afternoon or this morning was courtesy of Dave. So you got to give the man credit because he was important to this process. And happy frogging, everybody. Now for the test. No clapping yet. Okay, which one is this? Very good. Thank you, Chris. That was great. Growls. Listen for the growl. That's the Rio Grande leopard frog. All right, moving on. Now that my ace student back here already got it right. Yay. This one. All right, enough of that. Yeah, frog with very loud recording. So? Southern, right, the laughing frog. So everybody knows that the last one is going to be the plains leopard frog. Listen, now you just heard the southern, listen to the plains. 1, 2, 3, 1, 2, 3, 1, 2, 3. As opposed to They don't sound that much alike when you listen. All right. Thank you all very much for your time. I hope that you enjoyed it, and I hope you learned a little something. That was the whole goal. And let's go eat lunch.
Thank you. I want to present you with just a real quick little thank you. Here's our